And now today we're going to look at this from a slightly different angle, but kind of melding in together the clinical aspects of the disease along with the genetics of the disease. And we're going to be jumping into cell biology today. And the title of this lecture is discussing the non-cell autonomous components of ALS. And just these will be uploaded on Carmen, so you'll be able to see all of the slides. I just wanted to bring up a first paragraph here on cell biologists, great cell biologists of the 19th century, including Virchow, the German physician, widely known as the father of pathology, and the French physiologist Claude Bernard established this pivotal idea that individual cells function autonomously, and this is naturally, while being part of the whole organism. Since then, many pathological conditions, including all major neurodegenerative diseases, have traditionally been considered mechanistically cell autonomous, meaning that damage within a selective population of affected neurons alone suffices to produce a disease. That is, in the context of ALS, it's clinically manifesting as a motor neuron disease. Classically thought of this in the field of neurology textbooks as a motor neuron, a cell autonomous disease. Well, what do we mean by cell autonomous? This is a genetic trait in multicellular organisms in which only genotypically mutant cells exhibit the mutant phenotype. Conversely, a non-cell autonomous trait is one in which genotypically mutant cells cause other cells, regardless of their genotype, to exhibit a mutant phenotype. So we're going to dig into this concept here, and what ALS just less than a decade ago was thought to be a cell autonomous disease. And the field has really turned head over heels into thinking of this disease as a non-cell autonomous disease. Not necessarily immediately embraced by the community, but I think that we will show you, I'll show you hopefully in this lecture, many studies in which I do not have time to cover all of them, but really how ALS is truly a non-cell autonomous disorder. Now, just as a bit of background here, again on ALS, it's adult onset neurodegenerative disease, 20,000 individuals in the U.S., alone degeneration of motor neurons, in the cell autonomous aspect, paralysis, and ultimately death. We've talked about the sporadic versus the familial cases, and then many of these genes that have been identified within SOM1 mutations, 43 C9orf72 expansion gene. Vast majority, though, of the cases are still unknown. But the disease course is remarkably very similar. Whether you have the C9orf reading frame 72 expansion or a SOD1 mutation or a PDP43 mutation, for the most part, when clinicians see these patients, they cannot distinguish them. Maybe some of these frontotemporal dementia disorders are add-ons that would point you into looking at some of these diseases like C9orf reading frame 72 and PDP43 mutations that we've heard about. But they really look very similar as far as the clinical presentation. And what this is really kind of opening up the door of the questions here are there common mechanisms in all of these ALS cases? So again, progressive loss of motor neurons, and why I put this slide in is that really we have to think about the patient, there's severe muscle atrophy. The question is muscle involved in the course and progression of the disease? You've got the brain involved, there's inflammation within the motor cortex, there's loss of later 5 of the motor cortex, there's multiple regions of the spinal cord, there's the brainstem involved. Are these places, and what are the cell types that are involved in the disease? You've heard about many of the clinical features of loss of the motor neurons, which is found by electrophysiology, and it's also seen post-mortem at pathology analysis. There's massive motor neuron loss that occurs in this disease. Actually, things kind of point, and we'll dig into this a little bit later when we're thinking about the mechanisms, on the aspect that ALS has multiple clinical presentations, from asymmetric patterns of muscle weakness. And it can spread very quickly. It can be on one side for a period of time, switch to the other side as far as muscle paralysis, and then ultimately progress. Diagnosis is the involvement of both upper and lower motor neuron degeneration. And you exclude other disorders like MS and such, and then you get ultimately diagnosed by exclusion of other disorders and inclusion of muscle weakness followed by electrophysiological natures in this disease. We've heard there's no cure for the disease. There's symptomatic treatment and palliative care. Riluzole is only FDA-approved drug. Again, just thinking about the different types here, sporadic, 90% of ALS cases are familial, SOD1, GP, fused in sarcoma and C9orf72. Wanted to dig just, Dr. Burgess did a really nice job of, for the most part, saying that wherever within the SOD1 mutation that you have, within the SOD1 gene that you have a mutation, it's quite predictive potentially of developing ALS at some point in your life. And we talked a bit, Dr. Burgess talked about toxic gain of function, and I wanted to just refresh you on this because within the field, and you'll hear more about this from Dr. Beatty's lecture where she will be talking about many of the different models that we utilize within the field to study ALS. But I wanted to refresh on one because this is the model in which, or the mutation that the field has really spent most of their time studying. And we have mice as well as rats that include the mutation of SOD1. These are the human SOD1 genes that have been placed transgenically in the mice. The most commonly used mouse model is the SOD G93A, high copy number of SOD G93A mouse. And this is the human mutation of SOD1 gene fragment that's put into the animals at multiple copies. It's highly progressive, disease onset approximately 90 days of age, in stage approximately 130 days. There's another mouse that is routinely utilized, and it's the SOD G37R mice. What Don Cleveland's laboratory, and this will be important to discuss in the context of how we started to enter this area of non-cell autonomy. And some of the most convincing proof of non-cell autonomy was with the SOD G37R mouse. Now, this is what they did is to flox the human unit SOD1 gene that had the G37R mutation. Now, again, there's multiple copies, and there's LOXP sites flanking each of those copies. So it's not that after recombination that one removes the entire SOD1 mutation, but upon cream-mediated recombination, you selectively reduce SOD1 levels. Not necessarily 100%, but they're reducing the levels of SOD1. So these animals have a slower disease onset. So they have a different mutation within SOD1, G37R. These animals develop disease onset at 197 days of age, with end-stage approximately at 400 days of life. So with this concept of cell autonomy versus non-cell autonomy, some of the most convincing studies came out of Don Cleveland's laboratory at UC San Diego. And he took these flox animals. So the LOXP sites are quite unique on the aspect that you can add a pre-recombinase and induce a recombination event that selectively gets rid of whatever is in between the LOXP sites. So again, all of the mutant SOD1s have LOXP sites. Upon recombination, you're selectively removing and reducing the SOD1 in particular cell types. So what Cleveland's laboratory did was to generate and utilize Cree driver mice. So transgenic mice in which the Cree was under specific promoters, from motor neuron specific promoters to astrocyte and microglial specific promoters. And what they did then was to cross these flox animals with these Cree-specific drivers, motor neuron, astrocyte, oligodendrocyte, to selectively reduce the levels of mutant SOD1 in specific cell types and add Does this influence disease onset? Does this influence disease progression and survival of the animals? And what was published by this group was that transgenic reduction of mutant SOD1 in various cell types improved survival in all of the cases. So in the blue lines are the G37 animals that were the control animals, dying at approximately at the 300 days median survival. These are Kaplan-Meier survival curves. So 100% of animals are alive, 0% of the animals are alive. Upon the Cre-mediated recombination, this islet-1 Cre, this is selectively reducing mutant SOD1 in the motor neuron pool. So it's a driver for motor neurons. And they saw an extension, about a 20-day to 30-day extension of survival of these animals. More interestingly, when they reduced mutant SOD1 in glial fibrillary acidic protein driver Cre animals. So this was an astrocytic line. GFAP is expressed in astrocytes. This promoter selectively reduced SOD1 in astrocytes, and they found about a 50 to 60-day increase in survival. So striking right there, because here I just said this is a motor neuron disease, and we all grew up reading our textbooks. It's a motor neuron disease. But when you selectively ablate the SOD1 only in motor neurons, you get a 20-day increase of survival. And I just told you a 50 to 60-day increase of survival. When you selectively reduce SOD1 in an astrocyte, that was the first convincing proof to the field that, my goodness, another cell other than astrocytes might be involved. Now, there were other studies that I will not talk about of making mosaic animals that kind of got these studies off, and that is mixing wild-type cells into transgenic animals. We don't have time to go through this in this lecture, but if there's interest, I can certainly point individuals to what really catapulted these specific molecular biology transgenic studies. Now, in EVA1 Cre across animals, EVA1 is a promoter that is expressed predominantly in microglial cells, microglia and macrophages. And that had a striking increase of survival, where animals had a median increase of survival between 80 to 100 days. So microglia gave it. Reducing SOD1 in microglia resulted in the longest extension of survival that had really been seen in these ALS animals, with astrocytes coming next. And then shockingly to many within the field, that motor neurons weren't the primary culprit, at least on disease progression and survival of these animals. So what we've done here is to look at some of the historical survival data in ALS animals. And most things are published, and we'll talk later when we're talking about therapeutics in the course as far as what is a meaningful result in these mouse lines. And the field has some issues of things only increasing survival by 7 to 10 days. But here we had, with islet-1 Cre, a 21% increase of survival. CD11B had a 27% increase of survival. And this is looking at the extension of survival from disease onset. So what these studies showed was that reducing SOD1 in the motor neuron itself delayed disease onset. These other cell types, astrocytes and microglial cells, actually resulted in a slowing down of disease progression. So the problem of trying to stop onset is that most people show up to the clinician's office following onset. So it's almost as if that bird has already left. And what you have to actually do is try to slow down disease progression. And remarkably, it was these other cell types, not motor neurons, that were involved in the speed that ALS really progressed. So this is kind of more of a schematic overview of showing where mutant SOD1 contributors of SOD1 and what they meant in each of the various cell types. So in motor neurons here, mutant SOD1 actually accelerates onset and early disease. In astrocytes, by the reduction of mutant SOD1 is implicated to accelerate the disease progression. In microglia, it highly accelerates disease progression. New work that has come out from Jeff Rothstein's laboratory is showing that oligodendrocytes have also been involved and that they accelerate disease onset from mutant SOD1 within at least the NG2 positive, an early precursor of oligodendrocytes. So oligodendrocytes appear to be involved not only in onset, but also progression of the disease. Despite muscle having significant atrophy in the mouse model of the disease, selective ablation of SOD1 in these cells had zero effect on disease onset or progression in the mouse model of the disease. Also, there's microvascular hemorrhages that occur both in the mouse model of the disease as well as in humans. You find these microhemorrhages throughout the brain and spinal cord of these patients. So that might implicate that SOD1 was involved as a potential vascular disease. So what Cleveland's group also did was to selectively reduce SOD1 in those vascular endothelial cells, and there was zero effect on the disease. So the interpretation here is mutant SOD1 has no effect in endothelia. So this really opened up the door now of the complexities of the disease. If you haven't already appreciated the complexities of the genetics component, here now we're throwing in the complexities of the cellular components that are contributing to disease onset and disease progression. So Dr. Burgess, this is a highly complex slide here, but Dr. Burgess really put this very well 2 courses ago. And that is, if you can almost imagine a cellular process changing in ALS, you're right. So we hear many times that whatever you're studying in ALS, you're studying the right thing. Because if you can almost dream about it happening, pick your favorite organ out. Mitochondria. There's significant problems occurring in the mitochondria. Is there endoplasmic stress? Reticulum stress, absolutely, in multiple cell types. Is there a change in glutamate receptors and glutamate toxicities? It's happening in ALS. Is there proteasome inhibition? Absolutely. Are there synaptic vesicle defects, altered axonal transports, toxic extracellular mutant SOD1 spilling out from cells, which isn't good for cells? So the proposed mechanisms of toxicity in at least only SOD1-mediated ALS, excitotoxicity is the hyperactivation of motor neurons resulting from the failure to remove neurotransmitters glutamate from synapses due to the deficiency of glutamate transporters in these mice, along with this host. And that was really why I put the glutamate there. This has been something that has been studied the most in ALS, is this glutamate toxicity hypothesis. And actually, our only one FDA-approved drug is thought to reduce glutamate toxicity. It doesn't do very well on extending life in patients, about 3-month extension of survival in patients, which isn't necessarily something that's great, but that's where we are today as far as FDA-approved drugs. And then you have this whole host and slew of other issues that are occurring in the cell from multiple cell types. Again, motor neurons and disease onset, astrocytes for whatever reason driving progression. Microglial cells, these cells that are thought to be the scavenging cells, kind of the immune cell of our central nervous system, they're becoming hyperactivated. The astrocyte is the most numerous cell type in the brain. Greek for glue. We thought that they were just supposed to hold everything together, and now we're understanding the vast complexities of these astrocytic cells within the brain and spinal cord. So, do we think that all of these things are right? All of these things wrong? We're going to have a debate in a couple of weeks where you're going to be joining this debate with Arthur Burgess and I, and we're going to try to dig in. And this is a perfect slide for you to be thinking about and reviewing, because what is it about these problems that are going on in the different cell types, and what is this telling us to try to find an answer to the disease? Should we study one or all or even more of these pathways that are going wrong in the disease to come up with an answer? And I think this will make for a very good debate, because individuals within the field are focusing on their favorite box, and some people are thinking more generally. Are we thinking about this all wrong? So put your thinking caps on, and it will be a nice hot debate between Dr. Burgess and myself. So is this an aspect, this whole non-cell autonomy? Do we see this, is this solely an ALS freakish event? Or do we see this in other disease types? And I think that many of the neurodegenerative diseases and even cancer has considered and are embracing this concept that it might not be just one cell type, that there are other cell types involved within the course of the disease. And we can certainly, what others have done is to break this down in a nice review by Kristalinas within the Cleveland group, which in the Cleveland group at the time of writing this, and really showing that neurons are the target for many of these cells, but then astrocytes either haven't been tested in Alzheimer's disease, but certainly within Parkinson's disease has been implicated. Microglial cells, Schwann cells, and oligodendrocytes have also been implicated. So this isn't something very unique for just ALS. An understanding of cell biology is really emerging among multiple of these diseases. So all of these studies have really been focused on the mouse models of the disease instructing us. And I think that mouse models are extremely effective at studying different disease mechanisms, pathogenesis, and cell biology. But is this relevant? And I think that we'll hear later in the models development and then in the therapeutics talk of our course, how predictive are these models for ALS? And I think that might be part of our debate session that we have. But there's been a number of studies to implicate the non-cell autonomy aspects within ALS. Again, this sounds quite nice as I'm talking about this, but this was not well accepted within the field as being part of human ALS. And actually, there's still some debate, but I think the field has shifted now to think of ALS as a non-cell autonomous disease. So earlier I showed you mutant SOD1 within various cellular components causes ALS or is implicated in disease onset and progression in the mouse model. Is this true in a human cell? So what multiple groups did, and showing some of Brett Gage's work at the Salk Institute, what they did was the same experiment in a tissue culture dish with human cells. So they took a human astrocyte and introduced the mutant SOD1 into this astrocyte, and then did this co-culture. And the next slide we'll talk a bit more about these co-cultures. But was the first time, was one of the first papers to implicate that mutant SOD1 in a human astrocyte could induce motor neurons to die. So what are these co-culture-based models? Here we're going back to the studies of the mouse cells, in which mouse SOD1 mutant astrocytes were taken out of the mouse and cultured in a dish. And then motor neurons that were isolated from mouse embryonic stem cell derived motor neurons. So these are motor neurons that we can purify because we put in a motor neuron-specific promoter with a GMP. So we can enrich for motor neurons to watch these motor neurons and do this co-culture on top of the ALS astrocytes. And ALS astrocytes induce motor neurons to die. Very predictably, almost every single motor neuron will die in that tissue culture dish. We've taken similar approaches in our laboratory of deriving neural stem cells from animals where we can make oligodendrocytes, astrocytes, to try to also study these aspects of the non-cellular autonomy. And can we dig into the astrocytes from a cell biological point of view? So So here we can derive neural progenitor cells from the spinal cord, pass them, or dissociate the spinal cord, and go through these Purkinje gradients to isolate these neural progenitor cells, which we can grow them up to large amounts of cells. These neural progenitor cells can be induced into neurons, they can be pushed with IGF-1, for example, in dendrocytes, and then with ectopic administration of fetal bovine serum or BMP2, we can push them into astrocytes. We can then make motor neurons from these normal embryonic stem cells, mouse embryonic stem cells, utilizing knowledge from Columbia University, Tom Jessel's laboratory, where we can understand motor neuron specification genes to induce these motor neurons, to induce these embryonic stem cells to turn into motor neurons. We can prove that they're motor neurons by transplanting them into chick embryos, developing chick embryos, where these mouse embryonic stem cells will migrate down, turn on all of the motor neuron specification genes, and even project out to the axial muscles of these developing chicks. So we can make motor neurons for these co-culturing assays. What do these co-culture assays look like? Pardon me. So here are these co-cultures. You're looking at pseudo-colored motor neurons. So the green motor neurons have been turned black. You're looking at them on top of non-ALS derived astrocytes or the G93A mouse-derived mutant SOD1 astrocytes. And one day after the motor neurons are co-cultured, they look just fine. But things are different at 70, as early as 72 hours. Motor neurons are branching out here on top of the normal astrocytes, looking just fine, and they're shrinking, and many of them are disappearing on top of the ALS-derived astrocytes, and things just get worse for the motor neurons exposed to the astrocytes. Remember, this is a normal motor neuron, not an ALS motor neuron, but a normal motor neuron on top of an ALS astrocyte, and those astrocytes have the propensity and capacity to kill. We can look at this over time, and you can see a dramatic decline in the number of surviving motor neurons when exposed to these ALS astrocytes. So we have this nice assay. But once again, now we're looking at only 2% of all ALS cases. What about the sporadic ALS cases? We'll hear more about the iPS technologies used with bone stem cells in a later lecture. But some of the work that we have been doing, or me in my past life of a graduate student and postdoc at the Salk Institute, was part of a team to isolate neural progenitor cells from postmortem brains. So that is from dead people. We could mine out, even after 150 hours, living neural progenitor cells that we could then instruct into neurons, astrocytes, and oligodendrocytes. So we applied the same principle on ALS patients or non-ALS patients, deriving their spinal cord. So everything that I had just said back then on the first studies were taking a normal human cell and having to introduce the mutant SOD1. That's not necessarily naturally what happens in ALS, where you have a spontaneous mutation or a genetic mutation of SOD1. So this was our attempt at studying this for the first time in human ALS, known mutations in sporadic patients. So we isolated the progenitor cells and could make these into neurons, oligodendrocytes, or astrocytes. Just as a case in point, we'll dig into the astrocytes here. And are we making astrocytes from these cells? The answer is, when we take these neural progenitor cells and push them into astrocytes, they upregulate these astrocyte-enriched genes, which are here on the bottom, and now we produce astrocytes from these patients. So really individualized medicine, or studying this ALS individual. Fortunately, they passed away, but now this was the first time that we've studied an astrocyte from that patient. So what happens when we have motor neurons co-cultured, again, normal motor neurons on top of either non-ALS derived astrocytes, again, the human SOD1 mutation. Dr. Burgess mentioned the most common North American SOD1 mutation. Does anyone remember? Most common North American SOD1 mutation. One base pair change in SOD1. A4B mutation. A4B mutation is the most common North American mutation that causes the SOD1 mediated ALS. Is it a fast or slow progression? You've got a 50-50 chance here. Okay, somebody's got to give me. It's an A4B SOD1 mutation, fast or slow. And it's a very fast progression. It's actually one of the fastest ALS progressions with about a year and a half to 2-year median survival of these patients, where it's typically 4 to 5 years on other SOD1 mutations. So sporadic ALS is, we also have patients that had sporadic ALS. What that means is that despite sequencing, we couldn't find a mutation in any of the known mutations that have been described for ALS. So motor neurons are cultured. They look fine in 24 hours. They're spreading out and looking just fine on top of the non-ALS derived astrocytes, and not so good when the motor neurons, again, normal motor neurons on top of these human-derived samples. This was the first time that anyone had studied a human ALS patient's astrocyte in a dish with an A4B SOD1 mutation with sporadic ALS, and it was exactly as the mouse had predicted, that the astrocytes were indeed toxic and sent a toxic signal or a direct cell biology mechanism to kill these motor neurons. So one of the things that, as we dig into ALS, we had an opportunity here of testing the SOD1 mutation. That is, and we'll talk more in this course about lessening SOD1 mutation in the known genetic cases of SOD1 mutation. Well, we have this familial ALS SOD1 indication to test a suppression approach. But there's also a very unique finding within the field that SOD1, despite it accounting for 2% of all ALS cases with a known mutation, there's a multitude of laboratories that say SOD1 gets misfolded in not only the SOD1 mutations, but also sporadic mutations. This is 50-50 in the field. It's about 50% of people buy into this camp that SOD1 gets misfolded, 50% are still questioning whether that's a correct answer. But here was our opportunity to really address whether SOD1 might be a converging target in not only the SOD1 mutations, but also sporadic ALS, where individuals were showing misfolding of SOD1 was occurring. So here was our samples. We have control non-ALS, our A4B SOD1 in that 4944 in the red, and then sporadic ALS. This is looking at, on the top line, you basically see that motor neurons are surviving quite nicely on top of our control here. On the bottom is when we reduced SOD1 with a lentiviral vector expressing an shRNA against SOD1. So we had the ability to suppress SOD1 within these cells, and we had about 50% in all of the lines reduction of SOD1. So this was in the astrocytes performed co-culture again. Motor neurons are doing just fine when we suppress SOD1. In the case of where we know the genetic target, reducing SOD1 inside of the mutant SOD1 astrocytes was fully protective towards the motor neurons. Okay, that's where mutation. What about now these cases? And we have not full protection, but very good protection of reducing SOD1 in the sporadic cases. So that maybe we're now leaning on the 50% that says SOD1 is a target in more than just the mutant SOD1 cases. So intriguing results here for us to be thinking about. What I wanted to do was to also show you within science, sometimes despite us being supra excited about these results and modeling ALS for the first time, modeling human ALS astrocytes in tissue of culture dish for the first time in the world, where we can now show that astrocytes are implicated in all of the ALS cases that we've seen. we were excited, but the field basically said, okay, William Kaspar's group, yes, this is interesting, but you're studying damaged neural stem cells. And you're taking these neural stem cells from the inflamed spinal cords of the sickest of patients in which they were dead. So you're really not studying true ALS. So what the good question is, what is true ALS? I think what they were doing in the critique to us was to say, your neural stem cells have come from a toxic environment, from an area where the spinal cord has undergone microhemorrhages, inflammation. Your neural stem cells have been intrinsically damaged coming from an ALS patient. So if you think you're studying true ALS and this astrocyte coming from a neural stem cell is really telling us anything about ALS, you're kidding. You're kidding yourselves, is what the critique was. Stand up.
But aren't your controls from the same type of source, though?
The controls are from the same source, but not being exposed. So the question is it your controls from the same source of material post-mortem? Yes and no. Yes, it's coming from post-mortem within the same time period of isolation. But ALS has many things going on. Individuals might not be breathing as well, particularly as they get closer to their death. There's massive inflammation. Your neural stem cells are actually exposed to a much more inflammatory cytokine environment than in the other non-ALS cases. So is this just a collection of a cell sitting in a bad environment for a long period of time? So there's other ways in which of trying to derive this that the field was working on, including ourselves, and that is to use a non-central nervous system derived tissue to derive your astrocytes. And this is this concept of induced pluripotent stem cell technology, where patients' skin samples are isolated, grown in a tissue culture dish, reprogrammed. You'll hear about this in a subsequent class, so we'll really dig into the induced pluripotent stem cells to study these diseases. But you reprogram them with these factors and isolate these induced pluripotent stem cell colonies where you can then derive neurons, astrocytes, and other cell types for studying in either high content or doing cell biology studies. And basically this is a concept that we and others have developed of taking fibroblasts, putting in these transcription factors which will induce reprogramming to an embryonic-like stem cell from a patient's skin cell. Again, this won several years ago Science's prize of the breakthrough science of the year, of reprogramming a skin fibroblast into a cell that looked like an embryonic stem cell. So instead of going all the way towards an embryonic stem cell-like cell, or induced pluripotent stem cell, we derive the approach or utilize the approach of partial reprogramming, and that is these factors reduce the amount of fibroblast genes turning on the embryonic-like stem cell production, but you don't have to go all the way back to an embryonic stem cell-like. We will go into an intermediate, and what the field is called is an unstable intermediate of direct reprogramming of fibroblasts and neural degenerative cells. So there's advantages of doing this. One is it's much faster. You don't have to do clonal selection because all of the cells will respond to this within your tissue culture dish, and the maintenance is much less time consuming. And it's a very straightforward process of deriving neurons, astrocytes, and oligodendrocytes. Dr. Kolb will be talking to us about the RNA aspects of this disease. So you heard the TDP43, the fused sarcoma, the C9 open reading frame 72, all screaming at potentially RNA splicing and an RNA involvement of the disease. He'll be talking about this, what Dr. Cole does is to see many ALS patients in the clinic where he can derive patient skin fibroblasts, not from postmortem patients, but from patients who are being seen throughout the course of their disease. So we can actually study living ALS patients, and we'll show you some of those data today. And if I take a skin cell from the periphery, it hasn't been exposed to that bad environment of the spinal cord, the hypoxia, the inflammatory cytokines. This is a non-CNS tissue that's derived here. So within the process here, we can take a fibroblast from a patient, reprogram them with the OCT3, CMIC, SOX2, KLF4, and push them on the way to the induced pluripotent stem cell. But instead of going all that distance, you can change them into a neural progenitor cell media and get them into this unstable intermediate. It's actually then turns stable on the aspect of it's a neural stem cell that one can propagate. So they can express all of the nestin and PAX6 that should be expressed for a neural stem cell. But then one can push these cells into an astrocyte, limentin and GFAP. So these are markers for astrocytes. And they lose the fibroblast or collagen expression. So what do these cells look like now? We can grow them here they start at day 0. By as early as about 3 weeks into the process, we can make these patient-specific neural stem cells that we can make an astrocyte out of. They grow as neural spheres. We can also grow them as monolayers. But then importantly, we can push them into astrocyte marking cells after differentiating. So these neural stem cells generated within 3 weeks from patients can turn into an astrocyte for a study in the dish. They can also turn into oligodendrocytes. So what happens now when we take these cells? So the person who did this was Catherine Meyer, who spoke to you last week on C9 open reading grade 7, who really pioneered this work in our laboratory, where she took either in green are the normal patients, so they don't have ALS, A4B SOD1 patients, and we have sporadic ALS in this purple color, and in blue are the C9 open reading grade 72 patients. So now we've rapidly been able to model the disease, include C9 open reading grade 72 in these samples, and look to see whether a skin-derived astrocyte, not exposed to hypoxia, not exposed to the inflammatory milieu of a dead person's spinal cord, would still induce the same toxicity. And the short answer is yes. In every single case, 100% of the cases, we've been able to say that this person had ALS, even when we blinded ourselves to these patients, ALS or non-ALS patients. And for example, in the black line, this is an A4B SOD1 patient skin fibroblast. Over here is a black, the black line is an A4B neural progenitor cell. That's a different patient. So the toxicity was almost identical between an A4B neural stem cell that was derived from the postmortem spinal cord versus a patient that was living from a fibroblast in a different person. Also, this was the first indication of the C9 open reading frame 72, yet another mutation. Those astrocytes still show the same toxicity. Again, going back, are we seeing common molecular pathways, common cellular and or common molecular and biochemical things going wrong with these cells to induce an unstable or non-supportive environment for the motor neuron? Interestingly, we've heard about SOD1 suppression. When we suppress SOD1 in these cells, like what I showed you earlier, SOD1 in the postmortem astrocytes increased survival, we saw the same in our fibroblast neuron that's reducing SOD1, unless it was C9 open reading frame 72 patients. Very striking result, because there it's starting to segregate from a genetic diversity of these patients. So that's telling us something as we put our thinking caps on from a cell biological point of view that maybe there are different mechanisms, or maybe there are different genetic mechanisms to end in the same result. Interestingly, even the TDP43 mutations in further work shows similar astrocyte-mediated toxicity. And this is quite interesting on the aspect that this is a patient that has an A4B sub. It's not a patient. It's a member of a family that has the A4B SOD1 mutation that is not showing the disease yet clinically. But when we take their fibroblasts and the astrocytes, we can show that those astrocytes are less supportive than non-ALS derived. So that does not bode well. We're not using this as a clinical prediction at this point, but we're certainly, that would not be a good result that I would be hoping for. So this, I think, is just demonstrating the potential to use now these patient-specific fibroblasts as showing that we can model ALS-derived astrocyte toxicity. We're in the process now of trying to correlate the degree of toxicity. Some of these lines are much more aggressive than others on killing, and many times this correlates to disease severity. So the patients that I can tell you that every single A4B SOD1 patient that has died within 1 to 2 years, those are our most aggressive astrocytes, whether they come from the postmortem derived spinal cord or from a fibroblast. So interesting days here on potentially using these patient-derived cells as disease predictors for patients on disease progression. So I think this is just showing how one might imagine in developing high content or high throughput drug screens with these assays, and the laboratory is working on deriving patient-specific cells, multiple patients with 384-well format to do the drug screening or gene screening of your favorite genes in these assays, at least in an astrocyte. So we've talked about astrocytes, and we've got a bit more time, so I wanted to just throw in a couple more cells because I hopefully have impressed upon you that both the mouse studies, the transgenic mouse studies, the cell biology mouse studies of an astrocyte shows that astrocytes are a non-cell autonomous component of disease progression, but also in the human cases. I think now within our mind, it's highly appreciated that astrocytes are involved in the disease, or microglia involved in the disease. And we and others, Stan Appel's group in Texas have studied microglia. Now these are going back to the mouse studies. And I just wanted to show you some of the things that we've been able to do within the laboratory and also potentially implicate microglia as a non-cell autonomous component in the disease. Here, microglia have been made red and motor neurons are green. What you're first looking at are normal mouse microglia co-cultured with the motor neurons. And you're looking at about 24 to 36 hours of these interactions. Nothing that exciting other than red cells kind of moving around on the survey. Now we've isolated, Ashley Frakes in the laboratory isolated microglia from ALS animals. So the G93A microglia, co-cultured them with motor neurons and did the same experiment. And I want you to pay particularly attention to this area. But those microglia are bouncing around faster. They'll move around faster. You'll see indications of tugging, hitting, pulling, and engulfing. So you just saw, just to go back there, that was a number to start, about 50% of the cells that died. What we have been able to dissect is the molecular mechanism for how these microglia go bad. So I think from a cell biology point of view, we know that these microglia go bad and turn on the classical immunological response, but they go out of control when they have ALS, and turn on NF-kappa B, which turns on these cells to go off and eat motor neurons. Again, those were normal motor neurons, so the microglia have gone bad, and they're going after the motor neuron. And this is what we can see here. Motor neurons are completely gone in the context when they're exposed to ALS SOD1 microglia. But if you inhibit this NF-kappa B pathway, which turns them into an activated microglia, you can prevent this process. And we've done some transgenic studies, and when you reduce the inflammation, now in the inflammatory component, or in microglia specifically, one has about a 30-day increase of survival in the mouse line of ALS. So this is something that's went from a tissue culture study into the animals to implicate microglia getting activated and being a cellular target that one can actually disrupt that process with either being SOD1 or NF-kappa B inhibition to actually result in an increase. Okay, our oligodendrocytes, I want to leave some room for questions, but are oligodendrocytes involved in the disease? We and others are working on oligodendrocytes biology. Oligodendrocytes have been shown in the mouse to be involved in the disease. We can make oligodendrocytes in a tissue culture dish, and what we're showing here are the oligodendrocytes of ALS mice, but the results are quite similar to human-derived ALS oligodendrocytes on the aspect that they are much less supportive towards motor neurons when cultured. And in fact, about 50% of them die. They start shrinking their processes. And interestingly, motor neurons, when exposed to oligodendrocytes from ALS mice, become hyperexcitable. Remember I told you earlier the cytotoxicity hypothesis, and this is something that we see in human ALS patients, hyperexcitability of the motor neurons. Normal motor neurons become hyperexcitable when exposed to ALS oligodendrocytes. So we've covered motor neurons, astrocytes, microglia, and now oligodendrocytes. And I just want to leave you on the last slide here, thinking again about all of these cells that are potentially going wrong and are involved in motor neuron disease onset as well as disease progression. And I'm happy to answer any questions. Is a very good question, and that is these patients typically develop later in life. They either have mutations or they have the ticking time bomb with the mutation, or they develop it sporadically. Why does this happen later? And then secondly, what's protecting the motor neurons or potentially delaying this process from going even faster? What we see in ALS, it's a good question on the aspect that what we don't know is whether it's a disease of aging, on the aspect that these mutations cause tiny little punches to the cells, and then eventually the cell is just tired and cannot protect itself anymore. Or is it a disease of aging, that the cell loses some capacity to fight itself, and then the disease strikes? What we do know is within ALS, there's a lot of compensatory mechanisms occurring on in the disease. And that is, you'll see an increased amount of neurotrophic factors throughout, within the CSF, throughout the entire course of the disease. And these trophic factors are trying to keep, we think, are trying to keep the motor neurons alive for as long as it can, until these cells basically break out of control, ultimately at the demise of the motor neuron. So there's many things going on from a compensatory good standpoint for the motor neuron. And there's also some very bad things. So it's not exactly yin and yang in this situation. So many things are occurring. Does that help you? It's not a full answer. But so why? We don't know is the answer. Because I think that we don't know whether it's a disease, does aging or the cellular time clock have anything to do with?
Well, when we get started with games, they could take a cell from a non-diseased person and turn it into not an embryonic cell, but like a reprogrammed cell who is still able to read and produce a response. So it seems like whatever toxin it is, always able to produce it.
And that would improve, or an indication that those astrocytes, once they turn into it, once the cell turns into an astrocyte, that they become bad. And I think that's a tissue culture, do things happen faster in tissue culture niche? And I don't think we know the answers to those questions.
Right, do you know anything about the involvement of NFKB pathway in the toxicity mediated by the astrocytes? I don't know what could be a sense. You try to target even the microglia, but you try to target even the astrocytes?
Yeah, so actually that's where these studies started, is that we see that astrocytes turn on a whole host of inflammatory cytokines, actually become activated. And NF-kappa B gets turned on. It's one of the highest pathways that gets turned on in an astrocyte, in an ALS astrocyte. And in fact, that's where we spend a lot of time, initial time, reducing NF-kappa B, which reduced the amount of inflammatory cytokines from those astrocytes, and it did absolutely zero, if anything, to protecting the motor neurons. If anything, it made things worse, because we also reduced probably some of the good things that were coming.
Now, when you're thinking about diagnosing ALS, when you're talking about exclusion of genetic diseases, if you're thinking about just kind of assay for diagnosis, do you have any idea whether or not astrocytes derived from patients with genetic diseases would not have those results?
Good question. If you derive an astrocyte from another disease, such as multiple sclerosis, would you see those astrocytes being non-supportive towards the motor neuron? We're in the process of trying to get other neurodegenerative diseases at the time. What I can say is that at least we've taken in Duchenne muscular dystrophy, which is not a motor neuron disease. It's a muscle disease. But there also are neuronal components to the disease as far as within the brain and cognitive problems. But you clearly have muscle atrophy in this disease. We took those patient fibroblasts, made them into an astrocyte, and determined in this muscle degenerative disease, would an astrocyte still be supportive? And the answer was yes. So within the neuromuscular space, Duchenne muscular dystrophy derived astrocytes were not toxic. We have to look at other diseases.
Right. Have you ever done a Coculture experiment? You put the motor neurons on top of the microglia. So they have to be physically interacting, touching for that response that to occur. Have you ever done it where you put normal motor neurons on normal microglia, but then maybe like the upper chamber laid out new microglia and see if the condition itself can do it? So you know what I'm asking?
It's a good experiment. I'll tell you what we did to answer that question. And this is, can you activate another cell type, a normal cell type, with conditioned media from another cell type? Or,
Yeah.
so the experiment that we've done is take in ALS-derived astrocyte conditioned media. So we know that they're producing inflammatory cytokines, and put that into motor neurons on top of a normal astrocyte. And that was not, the motor neurons were fine. Did we not let the experiment go long enough? Did we not have enough of the media? Because we know that there are inflammatory cytokines and other probably bad things in the media coming from the ALS-derived astrocytes that will kill the motor neuron faster if the motor neuron is just by itself. But the normal astrocytes were able to buffer whatever inflammatory cytokine and toxicity that might have been transferred.
Would it buffer condition media from the strong microglia?
So for the webcast, what Dr. Gutridge is asking, which is a very good question, is whether how much crosstalk actually occurs within ALS. And it's something that is being studied by multiple groups. That is, do the microglia, who are thought to come first, are they influencing the astrocytes to go bad? Or is it vice versa? The astrocytes are influencing the microglia to go bad. No one has done a very clear experiment to really be able to decipher the true crosstalk. What you can say, I think that our transgenic studies have shown us that merely suppressing mutant SOD1 in an astrocyte does not appear to significantly influence microglial activation from a pathological point of view.
Have you ever gotten conditioned media from an ALS motor neuron and put that in with the astrocytes to see whether or not it's something motor neurons are secreting that is either activating or not the astrocytes?
So are motor neurons secreting something within the disease? So an ALS motor neuron secreting something that would activate other cell types. We have not personally done that, to my knowledge, in our laboratory. I think that the one group that I would inform the class to pay attention to the manuscript is from Jean-Pierre Julien's group in Canada, the progranulin being secreted. And I believe that Pierre Julien has shown that that could be a potential of misfolded SOD1 through the progranulin pathway is actually a potential aspect for motor neurons triggering, mutant motor neurons triggering disease. We haven't done that, but with our postmortem derived astrocytes, getting rid of misfolded SOD1 in that patient's cells completely rescued. So I would suspect yes, but it's worthy of doing. And last question, Anna.
Sorry. For all of the cells involved in the disease, has anyone sequenced each cell type and seen what pathways are conserved in each of them that are either upregulated or downregulated?
So let me see. Has one looked at the expression patterns in each of the cell types that are implicated in the disease to then say, is there a common pathway amongst all of the cells that could point you into the direction? People have been sequencing astrocytes and microglia and motor neurons ad nauseum within this field, and there's a tremendous amount of data that has been collected on these various cell types. Have we just not collected enough, I think, is a question. What I can tell you about the status of the field, we have extensive knowledge bases now of the gene expression profile, at least in the mouse, and even some in the human patients' derived cells. Nothing has come out as a screaming bullet to say what is the trigger, and I think this is a good way, unplanned way to end the class, and I'm happy to answer any questions. But what is the trigger And what is the cell that triggers the whole cascade of events to happen in the various cell types, and then in the various mechanisms and pathways from cytotoxicity to mitochondria? And what is that silver bullet? And how do you go about finding that silver bullet that is the trigger that sends all of these cascade of events moving forward? That is the trillion-dollar question. And something that the field, and that's where, as you're thinking about your proposals, how can you utilize your minds to find that silver bullet that is that cascading investment that what you'd like to ask? And I think that the debate will really be helpful because we'll start thinking as a group and as a class really about how best does one think about this disease as we're gaining more and more knowledge throughout this course, is how we should go and attack it, or how you should go